Hey guys, I am back again another time to film another video. So today we're going to be looking at prokaryotes and eukaryotes, microscopes, and the history of microscopes and cells and everything, and who founded it, who did what. Okay, so when I say take notes, just take down those notes and I recommend you study those notes. Okay, so the objective of this video is to state the cell theory, distinguish between eukaryotes and prokaryotes, and describe how different types of microscopes work. We have our flashcards for that. I recommend you write down these flashcards. We're going to get to those soon. Also, anybody in the class, if you don't have flashcards, I have like extra, so you guys could definitely ask me for the ones I see in person. So, microscopes and the cell. In 1665, Robert Hooke. Write Hooke down. You don't need to know the first name most likely on the test, but write Hooke down, okay? Write it down. Hooke, Englishman, you need to know this for the test. Hooke, Englishman, used an early microscope to look at a thin slice of cork and plant material. Now, moving on to the next one. Anton van Leeuwenhoek. Leeuwenhoek, sorry if I'm butchering that name. Leeuwenhoek, write that down. Write the last name down. You're going to need to know the last name. Now, Leeuwenhoek used a single lens microscope to look at human mouth and water samples. What did he see? He saw one-celled organisms, some bacteria are one-celled organisms, called animalcules, little animals. Animalcules means little animals, okay? Remember that. In 1838, he was German, Schleiden concluded that all plants were made of cells. This right here begins to go towards the direction of cell theory, okay? So Schleiden, there's Schleiden and Schwann, there's Leeuwenhoek and Hooke. Schleiden, Schwann, Hooke, and Leeuwenhoek. They're very similar names because they're all from the similar places, so it should be easier to remember them. Hooke, Leeuwenhoek, Hooke and Leeuwenhoek, Schleiden and Schwann. Think of Sliden and Swan. Think of it like that in your head. Maybe that'll help. And Leeuwenhoek and Hooke. Maybe that'll help you. So, German geologist Theodor Schwann. Okay, you don't need to know the first name. Schwann stated that all animals were made of cells. Again, this is leading towards cell theory. German physician Rudolf Virchow, you also need to remember Virchow. Virchow concluded that new cells are created only from division of existing cells. This is how new cells are made, and this was proven. New cells are made from the division of other cells. And in cancer as well, if a cell, something goes wrong with it and it makes too much, and cancer can happen like that. So, now we're at cell theory. For cell theory, you need to write this down, study it well, keep it in your head, memorize it like it's the back of your hand. Cell theory, because this cell theory, this is going to be the basis that we build this entire year upon. Cell theory. Because we're going to be learning all about cells, biology, all that stuff. Memorize cell theory like it is your name, okay? So, I'm going to state cell theory now. All living things are made of cells, number 1. Number 2, cells are the basic unit of structure and function in living things. Number 3, you only need to remember 3 things. 3 things. New cells are produced from existing cells. See, this was proven. Virchow concluded that new cells make other cells by dividing. And in cell theory, it was proven that new cells are produced from existing cells. So Virchow's conclusion is in cell theory, okay? So I'm going to restate all that I just said. I explained it. Now I'm going to say it just simply, so you can quickly write it down. Hooke, Englishman. Write Englishman down because you're going to need to know this for the test. Hooke, Englishman, used an early microscope to look at thin slice of cork and plant material. Leeuwenhoek used single-lens microscope to look at human mouth and water samples. He saw one-celled organism called animalcules, little animals. That means little animals. Schleiden concluded that all plants were made of cells. Schwann stated that all animals were made of cells. Virchow concluded new cell Virchow concluded that new cells are made from division of existing cells. I'm going to repeat cell theory again. So who are the fathers of cell theory? Schleiden, Schwann, and Virchow. Those are the fathers of cell theory, okay? Because they made all these conclusions up. And guess what? Those conclusions are inside cell theory. So their conclusions are inside cell theory. They are the fathers. Remember that. Write it down. Piece of notebook, your table, your arm, I don't care where. Remember that. So I'm going to restate cell theory again. All living things are made of cells. Cells are the basic unit of structure and function in living things. Living things. Not non-living things, okay? Remember that. If you don't know that they are the basic structure and function in living things. If you don't know that, you're not going to pass the test. You need to know. You need to write it in your head, living things. And new cells are produced from existing cells. So, know all these names, know where they're from, and know cell theory. First part done. Now we flip the page on my notes. Okay, prokaryotes and eukaryotes. Write this down. You can write it in a Venn diagram. That is how I wrote it. So prokaryotes, they don't have a nuclear membrane. What is a nuclear membrane? Sorry, I had to go there for a second. But prokaryotes don't have a nuclear membrane. But what is a nuclear membrane? A nuclear membrane is literally it's like a shell that encloses the cell's nucleus. The nucleus, you need to know what the nucleus is, what it carries. What does the nucleus carry? You need to know this. The nucleus, it has DNA inside of it. The nucleus will have DNA inside of it. Know this. Write it down. I don't care where. So, the nucleus, right? It needs to have a membrane around it in eukaryotes. In prokaryotes, it doesn't have a membrane around it. So basically, the DNA are just floating around everywhere. That's how it is in prokaryotes. The DNA is just floating around everywhere. But in eukaryotes, there is a nuclear membrane. So, when there is a nuclear membrane, it's going to look like a little circle. It's going to look like a little circle when we looked in the microscopes during class. And if you see a picture of a microscope of a eukaryotic cell, you are going to see like something like this. Maybe it's different shape, maybe it's jagged, maybe it looks like a star, whatever. And then there's going to be a dot somewhere there. It could look like a star. It could look all wonky. It could look like this, whatever it may look like. There's going to be a dot there, and that is the nucleus in a eukaryotic cell. So, prokaryotes have a flagella. I hope I'm pronouncing that right. And they have pili. I have to go there for a second. Sorry. Flagella are long tail-like things that enables the cell, sorry, it enables it to swim. So, the pili is shorter than the flagella, but hair-like structures. And why does it have this? It has it for the adhesion. Adhesion means like sticking to something, to surfaces, and for transferring genetic material. You most likely won't need to know why it exists, but just in case you were wondering, I told you. So, flagella, tail of the cell, okay? So, disregard what I just said. When I say tail of the cell, is the tail of the bacteria, actually. But bacteria is single-celled organisms. So, there for a second, sorry. So, a flagella is like a long tail for bacterial mobility, okay? So the bacteria can swim around. Sperm also has flagella. So, it's like a little tail for it to move around. So it can swim around, okay? That's what the flagella is. That's it. It's a tail. So now we're going to move on to the pili. Pili on bacteria is like little hair-like structures, and it's mostly for adhesion to surfaces and for transferring genetic material. You probably won't need to know that, but just in case. So, eukaryotes. We're moving on to eukaryotes now. Yes, they have a nuclear membrane, okay? I said this, and I will say it again. They have a nuclear membrane. Remember this. And they don't have flagella, and they don't have pili. Now, moving on. Whew. So, electron microscopes can see virus, protein, atoms, and molecules. There is more that they can see, but this is just what I wrote down for now. Just study that. Light microscopes. They can see bacteria, mitochondria, cells, and human egg. Okay? Human egg, what is that? Humans don't lay eggs, obviously. Human eggs is inside the female body, inside of the uterus, and it's kind of like it carries. I'm going to explain it better in a second. Give me a... Okay, I need to word that better next time. So, human egg cells carry genetic material. They carry 23 chromosomes. We're going to learn this soon in meiosis and mitosis. But disregard this information. You don't need to know this. So, more. So now, moving on to microscopes. We did this today. So, microscopes. Most microscopes use lenses to magnify image. So now, moving on to light microscopes. We have our flashcards. I recommend you get some flashcards and you start writing this down. So compound or light microscopes. Compound and light microscopes, they can be used interchangeably. They magnify by bending light. You need to know this. Magnify by bending light. Magnify 1,000 times their size, and they can see bacteria, mitochondria, cells, and human egg. My lord. So, scanning electron microscope, or SEM for short, produce 3D images of cells. If you go search up scanning electron microscopes, the images that they produce, oh my god, they are so beautiful and so detailed and so amazing. I recommend you go search that up right now. And the specimen that you use doesn't need to be thin. So it doesn't need to be cut thinly. The specimen you use to look at, it doesn't need to be cut thinly. So, transmission electron microscopes. They are used to study cell structures and protein. Know this, write it down. They are used to study cell structures and protein, and the specimen you use must be cut thin. Okay? How do you cut it thin like that? They have a special machine that they use. You obviously can't cut it by hand into that thin slices. Eukaryotes, we went over this. Prokaryotes, we went over this. And electron microscopes. They reveal details 1,000 times smaller. This is similar to the light or compound microscope, but light microscopes and compound microscopes magnify 1,000 times their size, but electron microscopes reveal details 1,000 times smaller. So they are very similar. It can be confused between the two, but you need to study that because I know some of you are going to be confused between the two. I know I'm going to be confused between some of them. So, and very, very important to detail that you cannot miss. Very important detail. Electron microscopes. You can only look at non-living things. Non-living, not alive. You can look at something that's not living. You cannot look at something that's living. And they see viruses, protein, atom, and molecule. Viruses, not living. Protein, not living. Atom, not living. Molecule, not living. So, now we have finally come to labeling the microscope. So, I'm going to put an image right here, and I want you to study this image a lot. So, first of all, we have the ocular lens, the lens you look into. Then we have the whole thing that is the eyepiece. So where the ocular lens is, there should be something like the lens. Imagine this is a microscope, and this is the lens, and this is where the lens is, sort of. This is where the lens is. This is where it sits. You're looking into it. This right here, this is the eyepiece. Okay? So, I'm going to pull up a picture right here in the back. Then, we have the nose piece. We have the nose piece. You need to know this. And the nose piece, you go down. There are lenses. We call those objective lenses. So, we have two types of lenses, ocular and objective. They can be confusing. They both start with an O, but ocular, you look at it with your eye. You're looking at it. Objective, the ones that are on the microscope, and when you spin them, there are lenses there to magnify. So there's those right there are called objective lenses. Now, you need to know the slide. You need to know the stage, and you need to know the light source. So the light source is pretty simple. It's going to be at the bottom of the microscope. At the bottom of the microscope is a light. Obviously, there are more pieces, but you don't need to know all of them for the test. But if you're planning to be a scientist in the future, you will definitely need to know the microscope, like all of them. All of the labels, sorry. So, you need to know the slide, the stage, the objective lens, the ocular lens, the eyepiece, and the clips. So what are the clips? So, when today we slid our specimen onto the stage. The stage is the area where our specimen sits. Imagine this pencil right here is our specimen. It sits right here, and the clips are two little clips that's going to just hold it in place while we look. It's going to hold our specimen in place. Very simple, very simple. It's just going to hold our specimen in place. The clips, they're very self-explanatory. Okay. So, I'm going over this again. the eyepiece. The big thing. We're looking into it. The ocular lens. Right here, that's what we're looking into. We're looking into the ocular lens. So we go down the microscope. We have objective lenses. Ocular and objective lenses. Study these and don't confuse them. And that is all for today. Yep, that is all. Thank you so much for watching. I hope you have a great day, and I hope you get a great grade on your test.